Melanocytes and Melanogenesis.
Oculocutaneous albinism type 1 is a genetic disorder caused by mutations on the TYR gene, leading to reduced or absent tyrosinase activity. Subtypes: OCA1A, complete lack of melanin due to absent tyrosinase activity. Its manifestations are white skin and hair, light blue or reddish pink eyes, severe visual impairments, no pigmentation development. OCA1B, partial melanin production due to reduced tyrosinase activity. Its manifestations are light skin and hair appear with gradual pigmentation, minor visual deficits. Complications are high risk of skin cancer, visual impairments, and potential psychosocial challenges. Vivalism is a genetic disorder affecting the migration of melanoblasts during embryonic development. It's caused by mutation in genes such as KIT or SNAI2. Its manifestations are patches of white skin hair, especially on the forehead and trunk. Stable and congenital pattern doesn't progress. Red hair, genetic variants in the melanocortin 1 receptor, MC1R, result in red hair pigmentation. It's caused by a predominant production of pheomelanin, red-yellow pigment, instead of pheomelanin, brown-black pigment. Its manifestations are red hair, fair skin, and light eyes, increased sensitivity to UV radiation, and higher melanoma risk. Vitiligo is an autoimmune disease causing the progressive destruction of melanocytes, resulting in loss of pigmentation in patches of skin. It causes a combination of genetic, environmental, and autoimmune factors. Its manifestations are irregular white patches, often symmetrical, and may involve hair, eyebrows, and eyelashes. Phenylketonuria is a metabolic disorder caused by a deficiency of the enzyme phenylalanine hydroxylase, leading to elevated phenylalanine levels in the body. Accumulated phenylalanine competitively inhibits tyrosinase activity, reducing melanin production. Its manifestations are fair skin, blonde hair, and light eyes, severe neurological damage if not diagnosed and treated yearly with a special diet.
Now, we're going to talk about the embryology of melanocytes. Melanocytes originate from neural crest cells, specifically melanoblasts, which migrate to various locations after the neural tube closes, such as the basal layer of the epidermis and hair follicles. The development, survival, and migration of melanocyte precursors are regulated by several genes, including microphthalmia-associated transcription factor, better known as MITF, known as the master transcription regulator. Other genes are PAX3, SUX10, EDNRB, EDN, KIT, and SNAE2. Neural crest cells arise from the primitive neuroectoderm and undergo an epithelial to mesenchymal transition during their migratory process. Upon reaching their destination, such as the skin in the epidermal basal layer or hair follicle matrix, or the hair or uveal tract of the eye, leptomeninges, or inner ear, they undergo a reverse mesenchymal to epithelial transition. By the 10th week of development, dermal melanocytes begin to differentiate. At 50 days of gestation, melanocytes reach the interfollicular epidermis and hair follicles. By the 4th month, melanocytes start producing melanosomes, the organelles responsible for melanin production. At birth, melanocytes are mainly distributed in the head, neck, and presacral region, and will remain localized in the basal layer throughout life.
The life cycle of melanocytes involves several stages: specialization of melanoblasts, migration, proliferation, differentiation into melanocytes, maturation, acquisition of dendritic morphology, and transport of melanosomes to keratinocytes, leading to cell death. Melanocytes are identified by specific proteins such as tyrosinase TYR, TYRP1, TYRP2, PML17, MART1, and MITF. Microscopically, mature melanocytes are oval or spindle-shaped, smaller than keratinocytes, and contain melanosomes. They reside in the basal layer of the epidermis, forming a melanoepidermis, until where one melanocyte is associated with 30-40 keratinocytes, at a ratio of 1 to 10. Approximately 1,200 melanocytes per square millimeters exist in the skin, with little variation among race. Pigmentation varies due to the size and number of melanosomes and the amount of melanin. Darker skin has larger and more numerous melanosomes.
Pigmentation and biological impairments. The skin pigmentation is the result of the interaction between genetic, environmental, and evolutionary factors that affect the production and distribution of melanin, which is the pigment responsibility for the skin color. The main function is protection against ultraviolet radiation. UV radiation can be harmful to the human tissues, causing DNA mutation and increase the risk of the skin cancer. Variation on intensity of the melanocytes. In less exposed regions such as the abdomen or the soles of the feet, the activity of melanocytes is usually low. In areas exposed to the sun, such as the face and hands, melanocytes tend to be more active to protect the skin against UV radiation. We have two types of the melanocytes: secrets and continents. The secrets melanin production is a defense mechanism, and with the continents, these melanocytes store melanin inside without actually transferred to the keratinocytes. Human population. The ability of dark and light skin to handle the effects of UV radiation varies significantly due to difference in the amount, type, and distribution of melanin. Dark skin contains a high concentration of melanin, a type of brown to black toning melanin, which is extremely efficient at absorbing and dispersing UV radiation. The light skin contains less melanin and higher proportion of fair melanin, a type of yellow to reddish hue melanin, which is less efficient at absorbing UV radiation, but the main function allows the synthesis of the vitamin D.
Melanin. Melanin is a pigmented polymer that in humans stored in melanosomes but in animals stored in melanophores. It is used to camouflage and for extinction of oxidated free radicals generated by UVR. We have two types of melanin: eumelanin that are black and brown color, and pheomelanin that are yellow and red color. Pigmentation depends of melanogenic activity, that proportion of mature melanosomes and their distribution. For example, in white skin, we have melanosomes smaller and closer in groups, but in dark skin, we have melanosomes larger, darker, and distributed more evenly. Now we have the stage of melanization. There are 7 stages of melanization. First, we have the transcription of required proteins. Second, the biogenesis of melanosomes. Third, the selection of melanogenic proteins. 4, we have the transport of melanosomes. 5, we have the transfer of melanosomes to keratinocytes, the melanophagocytosis. 6, we have the degradation of melanosomes. And finally, we have the removal of melanin by desquamation. Melanin biosynthesis. To initiate this process, we need the amino acid tyrosine with the assistance of the enzyme tyrosinase. This enzyme activity is inhibited by dopamine and stabilized by tyrosine-related protein 1. Tyrosine-related protein 2 has a similar function to tyrosines, as it participates in the synthesis pathway of melanin. Pheuromelanin synthesis requires less tyrosinase activity compared to eumelanin, which explains its production under conditions of lower enzymatic stimulation. We start with the amino acid phenylalanine, which with the help of phenylalanine hydroxylase is converted into tyrosine. Tyrosine, with the help of tyrosinase, is converted into DOPA. DOPA, under the action of tyrosinase, becomes dopaquinone, which follow 2 possibility pathways. First, it reacts with cysteine, forming cysteine DOPA, which leads to benzodiazine derivatives, resulting in the production of phenylalanines. The second way, it produces dopacron, which with the help of TRP2 is converted into 5,6-dehydroxyindole 2-carboxylic acid, forming eumelanin.
Melanosomes. Melanin synthesis occurs in melanosomes, which are organelles that are related to lysosomes. There are 4 stages in the development of melanosomes. Pre-melanosomes stage 1 are small, round vesicles, with an amorphous matrix. Stage 2 melanosomes have an organized structure fibrillary matrix, mainly of the GP100 family. P-mil17 deposit and tyrosine is present, but pigment synthesis has not started, forming in the endoplasmic reticulum and they are incorporated into the melanosomes. For complete maturation of melanosomes, an increase in intramelanosomal pH from 5 to 6.8 is required, which is dependent of P protein. The beginning of melanin production takes place at stage 3, where the pigment is deposited on fibrillary protein. In the last stage, stage 4, the entire melanosome is filled with pigment.
Melanogenesis represents the mechanism leading to melanin synthesis, which takes place in melanosome. 2 families of melanosomes are produced. 0.3 micrometers and oval with tubular glycoprotein matrix, which produce a dark brown or black insoluble primer. Is the main type in people with dark skin and hair and is more efficient in photoprotection. And the pheomelanosome, which are small 0.7 micrometers and spherical, with more disorganized and large matrix, which produce a soluble red-yellow polymer formed by the conjugation of cysteine or glutathione, is predominantly found in individuals with light red hair, prototypes 1 and 2. In one skin tumors are most common. Melanocytes produce a melanin from the amino acid tyrosine through the action of the enzyme tyrosinase, which is a glycoprotein located in the melanosomal membrane with an internal transmembrane and cytoplasmatic domain. It is a copper-dependent enzyme that catalyzes the conversion of L-tyrosine to L-DOPA, initial step of melanogenesis, and therefore this enzyme limits the rate of total melanin synthesis. There are 2 tyrosinase-like proteins with 40% homologous amino acids: tyrosinase-related protein 1, TRP1, and tyrosinase-related protein 2, TRP2, which are also located in the membrane of melanosomes, although their precise function has not yet been elucidated. It is possible that TRP1 plays a role in tyrosinase activation and stabilization, as well as participating in melanosome synthesis, increasing the pheomelanin ratio and playing a role against oxidative stress due to its peroxidase effect.
Melanosomes. Melanin synthesis occurs in melanosomes, which are lysosome-related organelles. There are 4 stages in melanosome development. Pre-melanosome stage 1 are small, round vesicles with an amorphous matrix. Melanosomes in stage 2 have a structured fibrous matrix, mainly of the GP100 family, and tyrosine is present. But pigment synthesis has not yet begun. For full maturation of melanosomes, an increase in pH in the melanosomal environment from 5 to 6.8 is needed, which depends on the protein that acts as proton in melanoma membrane. The initiation of melanin production occurs in stage 3, where the pigment is deposited on Fahnbuch's protein. In the final stage, stage 4, the melanosome is filled with the pigment, and melanosomes also lose the enzymatic activity of tyrosine to finally be transported to keratinocytes. through cytokine elements.
Next, transport of myosin B is an essential motor protein in the intracellular transport of melanosomes. This protein walks along the actin filaments of the cytoskeleton, dragging melanosomes towards the dendritic process of melanocytes. RAB27A and melanophyllin test the protein act as adapters connecting myosin B to the melanosomes. RAB27A adapters to the melanosomes membrane, while melanophylin binds to the both RAB27A and myosin B, forming a protein complex that facilitates the directed movement of the melanosomes. Phagocytosis of melanosome by keratinocytes a multifactorial process. The transfer of melanosome from the melanocytes to keratinocytes is a complex process involving multiple molecular signals. The keratinocytes growth factor receptor KGFR and its ligand KGF play a crucial role in the activation of melanosome phagocytosis. Upon binding to KGFR and KGF induces an intracellular signaling cascade that promotes the internalization of the melanosome by keratinocytes. PAR2 protease activate receptor 2 PAR2 is another important player in this process. Activation of PAR2 by specific proteases triggers a series of events that facilitate melanosome phagocytosis. Upon reaching its final location, melanin becomes the main determinant of skin color, which, in addition to definite and important human phenotypic traits, plays a critical role in photoprotection due to its ability to absorb ultraviolet radiation. Grisel syndrome is a rare genetic disease characterized by hypopigmentation of the skin and the hair and may present other manifestations: silver sheen in the hair, immunodeficiency, neurological impairments, uncontrolled activation of the macrophage and lymphocytes. Diffuse pigment dilution occurs. There is a lack of transfer of melanosomes from melanocytes to nearby keratinocytes. What is tyrosine? Tyrosine is a key enzyme in the production of melanin, the pigment that colors our skin, hair, and eyes. This process, known as melanogenesis, protects our skin from damage caused by ultraviolet radiation. Factors influencing tyrosine activity. The image you present highlights several factors that can influence tyrosine activity and thus melanin production. Melanotrophin. This hormone stimulates melanin production, which explains why our skin darkens when we're exposed to the sun. Basic fibroblast growth factor. Also, its main function is related to tissue repair. It has also been observed to influence melanin production. Endothelin-1. This substance, which is produced in the endothelial cells of blood vessels, can also stimulate tyrosine activity. UV radiation. Exposure to ultraviolet rays from the sun is one of the main stimuli for melanin production. Propionmelanocortin and its products. The image also mentioned propionmelanocortin, a precursor protein for several important hormones including ACTH, beta endorphin, MSH, alpha, beta, and gamma. The main connection between POMC and tyrosine activity is established through MSH. As mentioned above, MSH is one of the cleavage products of POMC. MSH and tyrosine. MSH is a potent stimulator of melanogenesis by binding to its specific receptors of melanocytes and induces an intracellular signaling cascade that activates tyrosine. This enzyme in turn catalyzes the oxidation reactions, leading to melanin agents that increase intraplasma MEPC levels. First, we have the choleratoxin. Choleratoxin activates the GE's protein by ATP revolution, stimulating adenylyl cyclase and increasing MEPC. The second is forskolin. Forskolin is a natural compound that directly activates adenylyl cyclase, raising MEPC levels. The next one is dibutyryl MEPC, a synthetic analog that crosses membranes and activates MEPC-dependent proteins. And then we have the MSH, binds to the MC1R receptor in melanocytes, activates the GEs, and increases MEPC. These agents increase intracellular MEPC levels, which activate pKa. pKa phosphorylates CREV, enabling the interregulate the transcription of MITF. MITF then stimulates the production of melanogenic enzymes, leading to increased melanin synthesis.
Ultraviolet radiation is classified as a complete carcinogen because it's a mutagen and a non-specific damaging agent. It's also a tumor initiator and a tumor promoter, but we need to remember it's also the most important modifiable risk factor for skin cancer. Ultraviolet radiation exposure results in epidermal hyperplasia due to growth factor production in the skin, epidermal hyperkeratosis or tanning, a immunosuppressive state of the skin, and production of vitamin D, as we saw in past slides. The types of ultraviolet radiation are ultraviolet C radiation that is mainly absorbed by atmospheric ozone and oxygen, ultraviolet B radiation that goes from 290 to 350 nanometers and constitutes about 5 to 10% of ambient ultraviolet radiation. It causes direct DNA mutation and is mainly absorbed by the epidermis. Finally, we have ultraviolet A radiation that goes from 315 to 400 nanometers. It constitutes about 90 to 95% of ambient ultraviolet radiation and generates reactive oxygen species that damage DNA, RNA, proteins, and lipids in our bodies. This is also subdivided into ultraviolet A1 radiation that goes from 300 to 350 nanometers and ultraviolet A2 radiation that goes from 350 to 400 nanometers.